Here's a quick video to help you with the worksheet that accompanied our DNA, RNA, and protein synthesis mini lesson. Just kind of start with DNA replication, and we're given a strand. We know that DNA is complementary, so as long as we have one strand, we can build the other from it. So let's go ahead and write down the bases associated with the strand that was given. Okay, and then again, because we know that DNA is complementary, we can go ahead and write the complementary strand using the strand that we were given. Oops, those are backwards. Let me erase that. Find my eraser. Okay. This is not easy writing on this iPad. Okay. Now I will go ahead and change the color of my print so that we can build our DNA. But first, I'll just remind you of a couple of things. DNA is unzipped by a DNA helicase. It comes in and breaks the H bonds, and it's broken at the origin of replication, or the replication fork. So this area right here, the DNA is being split down the middle this direction into that fork. So the second step after it's broken, after those bonds are broken, is for the DNA to unwind, and then it'll open up and spread apart, and that's what we're seeing here. After that, we have these enzymes called DNA polymerase coming in, and they're going to come in and they are going to attach new nucleotide bases that are complementary to the existing strands. So it doesn't matter where you start, just try to get as many as you can. And so I'll start at the top. And so we're just writing the complementary bases. Okay, and then go ahead and do that to the other side, because this is occurring on both strands simultaneously. So I'm just using the complementary base pairing rules established by Chargaff to build my DNA strands. All right. And that's going to provide the backbone as well, because remember, a nucleotide is what's being brought in, and that consists of a sugar, the phosphate group, and the base. All right, and so if you were giving something like this, you would complete it as I showed you here, keeping in mind that we have two new identical strands, and it's referred to as semi-conservative replication because we have part of the old strand in the red. We call that the parent strand. And then we have the new strand, which is referred to as the daughter strand. And so it's semi-conservative in that we're using the old strand to make the new strand. All right? And that is DNA replication. This one is to help you with the protein synthesis part. And so we see that we're given a strand of DNA, and we have to undergo transcription, which is where we're going to make a copy of RNA using the DNA that we're given. And we have to remember when we do this that with RNA, we do not have T anymore. We have to replace T with U. So anywhere we would have placed a T, we're going to now use a U when we transcribe that. And this first mRNA that we're doing, this first RNA that we're doing is mRNA. It's the message from the DNA. So let me get my pencil going again. Sorry about that. I'm just trying to do this really quick. Okay, and then, so we have A. It would have bonded to T, except for its RNA, so we're going to get a U here. Sorry about this. But it's something other than what you do not have right now. That's an A. It's very small. So that's the first step of protein synthesis, and again, that's transcription. That is occurring in the nucleus where the DNA is. Once we have our mRNA strand, it is going to leave the nucleus and enter the cytoplasm, where it will be bonded to an rRNA molecule, the ribosome, for further processing. And now we're going to start with the translation piece. And so we use the mRNA because that's the message of DNA. And we're going to bring in some transfer RNAs that are carrying the amino acids. So the next step in our work is to identify the bases found in the tRNA. Remember that we use triplet codes when we're talking about RNA, and so in the mRNA, this is referred to as a codon. Oops, I don't know what's going on. So it's referred to as a codon, and then this part here on the tRNA is referred to as an anticodon. So that's what we're going to be identifying now, or decoding, if you will. All right, so let me get ready. And so again, this is an RNA strand that we're making now, still, so we are going to have to use U instead of T. Okay, and then the last, now that we have these, see these top tRNAs are bringing in the protein, or the amino acid I should say. It's bringing in the different amino acids to build the protein. And so we have to use actually the mRNA strand, because that's going to tell us what is being coded for the protein, for the amino acids. So I've got the table so that we can learn how to use the table or practice using the table. And we're going to use the mRNA strand. And so the first ones I have is UAG, so I find U, I find the A, and I find the G. And where all those come together, that is my amino acid. We cannot start building our protein until we get our start codon. That's a stop, so we're going to ignore that one and move to the next one. The next one is the UAG, because remember we're on our mRNA strand. Let me go ahead and erase these. It is not letting me erase. I'm not sure why this time. I'm going to try one more time and then move on. Okay, well, I won't be erasing those, so we'll just have to kind of ignore them. All right, so anyway, AUG. So we've got A. It's doing all kinds of weird things on my end. So we've got the A, UG. So there's A there, and then we got to take the U down to here to the G. So we got the MET. That's our start codon. That's one I told you to memorize. Methionine is always the start. So we can start writing our protein. And then we have ACC. So again, we're going to follow it over. A, C, until we get to the C. So THR. And then we have the AUA. So again, our A going to the U and the A. So ILE. And then we have CCA. So CCA. Oops, that one's not the A. This one's the A. They're all the same, so it didn't matter. But sorry about that jumbled up mess. Hopefully it's enough that you can see what we're doing and everything. All right, so this is one practice of protein synthesis. And that last piece where we were bringing in the proteins or building our protein from the amino acids, that is called translation. And remember, again, there's a little bond between each of these amino acids. And those bonds are called peptide bonds. Okay, I'm going to move on. AC. Oh, goodness. All right, so here's another sample problem similar to one you're going to find on your worksheet. And you see that you're given the DNA strand again. And you have to write out the mRNA and your tRNAs. And then identify the growing protein. So you need to identify the amino acids. This time I've given you a circle chart so that we can practice using the circle chart in the video. So because I'm making RNA, I do not use T's. I use U's instead. So we'll start by building our G, G, U, U, A, A.
Whatever party you dream up, Party City will help you bring it to life with thousands of new products in all the latest styles for birthdays, weddings, baby showers, and more. Party City has endless possibilities to make your party unique.
Okay, and then we're going to write the anticodons. Remember, the tRNA is the anticodon.
Your money-saving coupon, good in-store or online. Party City. Oh, it's on.
You have to be up in 5 hours. 2 hours. 6 minutes, and you haven't slept a week. ZQL Pure Z's is a drug-free blend of botanicals with an optimal dose of melatonin, so you can fall asleep naturally and wake with no next-day grogginess. So tonight, try new ZQL Pure Z's from the sleep experts at Fix ZQL.
Okay, almost done with that. Again, I'm just trying to put something together for you really quick to help you with this process for those of you who need it. So now I'm going to read it remember using the mRNA strand. Okay, that's what I have to translate. And this is occurring at the ribosomes and the cytoplasm. And so I look at my UCU, and the way we use these is we go from the inside out. So UCU would be my first pathway, and that's SER. Remember, I cannot begin building my protein and I get methionine or the start codon. So then I'm not going to write anything there. And then we got UGU, UGU, UGU. Again, it's not my start. And then I have AUG. So AUG. I finally have my start right here. So that's where I'm going to begin methionine. Then GAG. Now I'm going to continue to translate this until I get to a stop codon. So GAG, GAG, then AUU, AUU, UCU, UCU again, SER, GGU, GGU, then UAA, UAA. That's my stop. And we don't write anything for stop, we just stop. And there is our protein made from this strand of DNA. All right, hopefully that helped. Sorry for the long video. Use it or don't. It's all up to you guys. All right, thank you.